Hello guys, it's me again, Aaron Funa. So today, we are going to design primers for PCR amplification at the convenience of our own home. But before that, let's define first what is a primer. So, let's start.
A primer is a short single-stranded nucleic acid utilized by all living organisms in the initiation of DNA synthesis. The polymerase chain reaction or PCR uses a pair of custom primers to direct DNA elongation toward each other at opposite ends of the sequence being amplified. These primers are typically between 18 and 24 bases in length and must code for only the specific upstream and downstream sites of the sequence being amplified. A primer that can bind to multiple regions along the DNA will amplify them all, eliminating the purpose of PCR.
So you already know what primers are. Now, let's move on to designing our very own primers.
To start designing our primers, we need to go to the NCBI website. NCBI stands for National Center for Biotechnology Information. It advances science and health by providing access to biomedical and genomic information. For our case today, we are going to look for the nucleotide of phenylalanine hydroxylase, which is responsible for phenylketonuria. To give you some background about phenylketonuria or PKU, it is an autosomal recessive inborn error of phenylalanine metabolism resulting from deficiency of phenylalanine hydroxylase or PAH. Most forms of PKU and hyperphenylalaninemia are caused by mutations in the PAH gene on chromosome 12, long arm 23. Now, here's the results of our search. We have the literature, the genes, proteins, genomes, clinical, and PubChem relevant to phenylketonuria. In here, we want to go to the nucleotide section. As you can see, there are 40 items of Homo sapiens DNA relevant to phenylketonuria. What we want to do next is to filter the information for the mRNA of phenylalanine hydroxylase. From 20 items, we now have 14 items. We further filter the items to RefSeq only because RefSeq databases provide non-redundant curated data representing our current knowledge of known genes. From 14 items, we now have 4 items to choose from. In choosing, we have to consider the number of base pairs. So the largest numbers are in 1 and 2, transcript variant 2 and transcript variant 1. So we chose transcript variant 2 with 3,987 base pairs of linear messenger RNA. As you can see on the definition, it is for Homo sapiens or human phenylalanine hydroxylase, transcript variant 2, messenger RNA. It has accession number of NM_001354308304. You can actually search for the accession number to find the same file. References, authors, titles, journal, PubMed number, and remark are also listed here. You can also find here the summary of the gene. In our case, the PAH gene encodes a member of the biopterin-dependent aromatic amino acid hydroxylase protein family. The encoded phenylalanine hydroxylase enzyme hydroxylates phenylalanine to tyrosine and is the rate-limiting step in phenylalanine catabolism. Deficiency of this enzyme activity results in the autosomal recessive disorder phenylketonuria. The variant is also described. It is stated here that variant 2 differs in the 5' UTR compared to variant 1. Both variants 1 and 2 encode the same protein. Also, listed here are the parts of the gene wherein you can locate the specific part. If you click gene, it will give you the entire gene. And if you click the CDS or the coding sequence, it will give you only the part of the coding sequence. Right now, our goal is to amplify the coding sequence or to make copies of it. In order to do this, we have to extract the FASTA file first. After we have extracted the FASTA format, we are now going to click the Pick Primers. After we have set all the parameters in place, we are now going to click the Get Primers. We now have generated the primer blast results. The graphical view of primer pairs shows the exons, the coding systems or sequence, and the 10 best PCR primers generated. This is the primer 1, the primer 2, primer 4. As you can see, primer 4 is the longest among the generated primers, but it may not be the best primer for us to use. So, we need to look at the characteristics of each primer. What we need to look at are the completeness of the base pairs, the length, the temperature, the GC percentage, and the self-complementarity and self-3' complementarity. Considering the mentioned factors, we can now conclude that the best primer to be used is the primer pair 5. As you can see, the difference between forward primer and reverse primer in terms of temperature is low. The GC percentage of the two are high, and the self-complementarity and self-3' complementarity are low compared to other primer pairs. Our next goal is to compare the two primers to other species. If there are other species with the same primers we have generated from human. So in order to do this, we have to copy and paste the forward primer and the reverse primer. We will change refsec mRNA to refsec RNA, and then we will remove the codings for human or Homo sapiens to generate list of other species with the same primer sequence to our generated primers a while ago. So now, we have generated the list of different species having the same sequence or mRNA sequence of human phenylalanine hydroxylase variant 2. We have listed Chlorocebus sabaeus, Cebus imitator, Myotis myotis, Calitrix jacus, Pan paniscus, Tachypeticus françoisi, Rhinolophus ferumequinum, Sapahus apella, Hylobates molo, Pilocolubus tephrocelis, Gorilla gorilla, and many more. So basically, the primers that we have generated are not unique for human phenylalanine hydroxylase, but it can also be used for other species.
So that's it, guys. I hope you learned and liked our video. So now it is your turn to do your primers for PCR amplification. So thank you guys for watching. Peace out.